With all of this going on, it's almost hard to believe that DNA can be replicated with faithfulness to the original sequence. The error rate of DNA replication is less than one mistake per billion nucleotides. The reason is that DNA polymerase has high fidelity, meaning it will accurately copy the sequence into a complementary strand. Any error that DNA polymerase III makes when it is elongating a strand of DNA, anytime the wrong nucleotide base pairs with the nucleotide that's on the template strand, DNA polymerase III will recognize that and remove it, essentially hit the delete key, because it has a 3' to 5' exonuclease activity. It can proofread its work. If an incorrect base pair is detected, the incorrect base will be removed by the exonuclease activity, and then DNA polymerase III's 5' to 3' polymerase activity will add in the correct nucleotide. It's still not perfect though. DNA polymerase III will still place the incorrect nucleotide approximately once every 10 million nucleotides. And for that reason, all organisms have evolved a mismatch repair system. These are enzymes that can detect a mismatch, meaning an incorrectly base paired nucleotide, and remove that mismatch and repair it with the correct sequence. What these enzymes have to do is first, as I said, recognize the mismatch, and then they have to be able to determine which of the two strands has the correct sequence. The way that bacteria do this, distinguish between the original template strand and the newly synthesized strand, that's the one that needs to be repaired, is that the original DNA is methylated and the newly synthesized strand is not methylated. In this way, the repair enzymes know which of the two strands of that helix needs to be repaired. Eukaryotes have a different way of distinguishing between the original old strand and the new incorrect strand, and it seems that in eukaryotes, the mismatch repair system is recognizing which of the strands has nicks due to lagging strand synthesis of Okazaki fragments. And as long as it can be recognized quickly before those fragments are joined to each other, the nicks will help identify which of the strands the mismatch repair enzymes need to correct. Eukaryotes also have another difference from prokaryotes. We know that RNA primers are removed by DNA polymerase I and have to be replaced by DNA nucleotides. This isn't a problem for a circular molecule in a bacteria cell because there's always a 3' hydroxyl available for DNA polymerase I to add new DNA nucleotides to. But in eukaryotes, at the very end of our linear chromosomes, there is no 3' hydroxyl available. Once DNA polymerase I removes the primer RNAs, there is no 3' end to which it can add a new covalent bond to join a DNA nucleotide. And therefore, each round of replication, the DNA strands will get shorter and shorter at the ends of the linear chromosome, at the telomeres. Eventually, gene sequences that are near the ends of chromosomes will be eroded. The chromosome will become unstable, will become degraded, and eventually the cell's functions will be altered and the cell will die. This end problem is illustrated best in these figures from the Bio 1 Campbell textbook. Here we're looking at replication at the end of a chromosome. The parental strands are in dark blue, the newly synthesized strands are in light blue. If we blow up the very end of the lagging strand, what we see is that the very last fragment on that lagging strand begins with an RNA primer at the 5' end and has been extended with light blue DNA nucleotides. The next to last fragment begins with a red RNA primer, right, and then continues into light blue DNA. But when DNA polymerase 1 replaces those red, in this case, red RNA nucleotides with DNA nucleotides, it can do it fine for the next to last fragment, but not for the very last fragment. DNA polymerase 1 can remove the RNA nucleotides, but there is no 3' end to which it can add DNA nucleotides. And so you end up with what we call a 3' overhang on that DNA double helix. Then, in the next round of replication, the new leading strand that's synthesized will be shorter than the original, and the new lagging strand that will be synthesized will also have another 3' overhang. And so in each subsequent round of DNA replication, the lagging strands are always going to get shorter and shorter. And what are the ends of our chromosomes called? Those are our telomeres. But what do we know about the structure of our telomeres? They are heterochromatic regions. They consist of many short sequences that are repeated hundreds of times over and over again. And therefore, because none of those DNA sequences code for any genetic information, they will postpone the erosion of vital gene sequences. So if the telomeres become shorter during each round of replication, that's not a problem for the genetic information. They're sort of buffering the erosion of important sequences. But after many rounds of DNA replication and cell division, the telomeres will be eroded and some of the more internal and more important sequences for gene expression may become degraded. And therefore, the shortening of telomeres appears to contribute to the aging process. Most of the cells in our body, and these are non-germ cells, so the cells that make up our skin, our muscles, our heart, most of those cells have a genetically programmed lifespan. They can usually divide about 20 to 50 times before they're destined to die. Other cells, such as the germline cells that will produce gametes, cells that need to proliferate, generate new cells over and over again in the lifetime of the organism, like the cells that line our intestine, like the cells that give rise to all of the blood cells, have a particular process that prevents the shortening of telomeres. Single-celled organisms would have to do the same thing. They would have to have a way to prevent shortening of telomeres, which would result in the death of the organism. That enzyme is called telomerase. It can restore the length of a telomere. Telomerase is composed of protein as well as an RNA molecule that is complementary to the G-rich strand that is unique to the telomeric sequences of any given organism. In this figure, in part A, we're looking at the telomere of a single-celled organism called Tetrahymena. Notice that the repeating sequences of this telomere, remember they would be repeated hundreds to thousands of times, TT, GG, GG, extend as a 3' overhang. The telomerase for this organism consists of an enzyme that is associated with a specific RNA molecule, shown in green. Notice that that RNA is complementary to the telomeric sequences, and therefore it can bind to that 3' overhang. This RNA molecule in the telomerase then serves as a template so that that G-rich 3' overhang can be elongated. By the normal DNA replication process, DNA polymerase III will be able to add DNA nucleotides one at a time, extending that 3' overhang. Then the telomerase will move several times, repeat this process over and over again until it's added about 15 or more nucleotides to the 3' end. In this way, the telomerase activity compensates for the repeated shortening of the telomeric sequences that occurs during each round of DNA replication of the chromosome. And overall, the telomeres do not shorten. So what if we used telomerase as a therapy to counteract the shortening that occurs during normal DNA replication of linear chromosomes? Would we be able to extend the natural lifespan of our cells? Would we be able to live longer? Would we be able to live forever? There would be a problem with that. We know that about 90% of all cancer cells have active telomerase, which allows them to live and divide indefinitely. So if we expressed a telomerase in all of our cells, it's possible that they would continue to proliferate and become cancerous. Although telomerase itself alone wouldn't lead to cancerous growth, it would help any defective cells to be able to continue to proliferate.